everyone, my name is William G and I'll be presenting to you a section of a paper entitled Targeting of the Human Factor VIII at the Multi-Copy rDNA Locus in Hemophilia A Patient-Derived Induced Pluripotent Stem Cells using Telonucleases. And here are the authors of this paper. So as a general summary of the presentation that I'll be giving, so I'll be presenting a section of this paper. So in this section I'll be presenting what they did was they used Yamanaka transcription factors to produce induced pluripotent stem cells from samples or cells they collected from urine samples of hemophilia A patients. Hemophilia is an X-linked genetic disorder and patients with hemophilia, they're not able to clot their blood. As you see in this figure, this is in the blood vessel. If there's a hemorrhage in the blood vessel, in a healthy individual, it clots by itself because the individual has certain proteins to do so. However, in hemophiliac patients, when there's a hemorrhage within the blood vessel, it is not able to clot, or this efficiency of clotting is significantly decreased. The inability to clot is caused by mutation in the F8 gene, which codes or produces a protein called blood coagulation factor VIII. Patients with hemophilia express three stages of symptoms: the mild, moderate, and severe. Patients with mild symptoms, they have 5 to 30% of their factor VIII protein active, while in moderate, they only have 2 to 5% of their protein active, while in severe cases, less than 1% of their factor VIII proteins are active. And among hemophilia A patients, 50% show mild symptoms, 10% show moderate symptoms, and about 40% show severe symptoms. Here is a general or a basic mechanism of how factor VIII protein plays a role in blood clotting. Factor VIII over here, should be factor VIII, gets activated by a molecule called thrombin. Once it's activated, along with other calcium ions and phospholipids, it helps in the activation of factor X or factor 10. Once factor 10 is activated, as denoted as factor XA, A standing for activation, factor XA then changes prothrombin into thrombin, and then in which thrombin helps in the activation of another factor VIII. So basically, when factor VIII is absent within the system, it will not allow the body to clot, or it will significantly decrease in the ability to clot. The main objectives of the section I was presenting was they use Yamanaka transcription factors to generate induced pluripotent stem cells. As you see over here, Yamanaka transcription factors including KLF4, SOX2, CMYC, NANOG, OCT3 and 4, and Lin28. They use these transcription factors to reprogram the cells and to produce induced pluripotent stem cells from urine samples of hemophilia A patients. And they later use these iPSCs to create the differentiated cells. What they first did was they collected some urine samples from the patients, and they used the epithelial cells found in that urine sample, and they infected these epithelial cells with retroviral vectors that contain the genes for OCT4, SOX2, KLF4, and CMYC. Here's the basic mechanism of how a retrovirus works. Once the virus infects the host cell, since it has RNA as its genomic content, it has to go through reverse transcription to create cDNA or complementary DNA, which then gets incorporated or integrated into the host cell's genome. And being dependent on the cell cycle of the host cell, it produces viral RNA and then later on producing the viral proteins that are needed. These three pictures show the morphology of the hemophilia A patient-derived iPSCs. The first picture shows just the urine cells or the epithelial cells from the urine samples of the hemophilia A patients. And in the second picture, the small cell clump in the middle is shown, and it is at the very early stage after introducing the transcription factors. And the third picture over here, it is the expanded form of the iPSCs, and it was taken after around 20 days after introducing the Yamanaka transcription factors. They use a technique called immunofluorescence, and this is a technique to basically visualize localization of specific proteins or antigens within the cell. A first antibody binds to the antigen, and then the second antibody that has a fluorophore attached to it binds to the first antibody, and then this whole thing is visualized under fluorescence microscope. So using immunofluorescence, as you see, they were able to detect some different pluripotent markers, and they were able to detect 5 of them: OCT4, SSEA1, SSEA4, TRAL160, and TRAL181. As you see over here, the green denotes for OCT4, and then the red over here denotes for the SSEA1 and 4, and also the red over here detects TRAL160 and TRAL181. The API, it detects for the presence of DNA, and therefore it was used as a positive control, and it is shown in green. In blue. They also use reverse transcription and quantitative PCR. Reverse transcription is basically the same thing as PCR, it's just that they use RNA as their template, and therefore they need reverse transcriptase to produce cDNA or complementary DNA. And once double strand is produced, they use normal PCR analysis to amplify this specific sequence of DNA. Quantitative PCR, on the other hand, use fluorescent dyes that bind specifically to double-stranded DNAs, and it helps the researchers to quantitatively measure the DNA amplification process. So using reverse transcription PCR and quantitative PCR, they were able to detect the expression of endogenous genes, or the genes that are present within the cell, and they're also able to find out the silencing of the exogenous factors or genes that had been introduced from the outside source. As shown in this figure over here, they were able to detect the expression of OCT4, SOX2, NANOG, and HTERT, HTERT standing for the telomerase reverse transcriptase. So basically they were able to detect all these 4 in the induced pluripotent stem cells. However, they were able to detect the silencing of the exogenous factors OCT4, SOX2, KL4, and c-MYC, as shown in the graph over here. When they karyotyped this hemophilia A patient-derived induced pluripotent stem cells, they were able to find out that it had a normal karyotype, as you see in this figure, meaning that despite their pluripotency, or despite inducing their pluripotency, there was no big damage done to the genomic content of the cell. Furthermore, they didn't explain the whole protocol of this, but they were able to detect the 3 different germ layers from the hemophilia A patient-derived induced pluripotent stem cells. They were able to detect the ectoderm, mesoderm, and the endoderm. And this is the figure for the ectoderm, the middle figure for the mesoderm, and the last figure for the endoderm. So to conclude this portion of the paper that I was presenting, the researchers were basically able to generate the induced pluripotent stem cells, or iPSCs, from urine samples of hemophilia A patients by introducing Yamanaka transcription factors. As you see here, they introduced Yamanaka transcription factor using a retrovirus, and this retrovirus enabled the cell to be reprogrammed using those transcription factors to produce the pluripotent stem cell lines. And the researchers were also able to show that the hemophilia A patient-derived induced pluripotent stem cells also showed potential to differentiate into the 3 germ layers, which were the ectoderm, mesoderm, and the endoderm. For future directions, or for the next step, they used the transcription activator like effector nicases or talon nicases to target the factor 8 gene at the multi-copy ribosomal DNA, rDNA, which is shown here. So what they did was they inserted genes that would produce the factor 8 proteins to rescue the shortage of factor 8 protein. And here are my references.